Hello everyone to this iReceptor seminars series in November 2022. Today we have again two speakers. We have the more senior speaker, Katharina Imkeller, and then we have the early career speaker, Jared Schiltz. I will start by introducing Katharina. She's a group leader for computational immunology at the University Hospital in Frankfurt, Germany. And we are very much looking forward to your talk today.
Yeah, hi everyone. Thank you, Victor, for the introduction, and thank you for the organizing team to invite me to speak today. As Victor mentioned, I have a group in computational immunology. We're located in Frankfurt am Main in Germany. And I would like to talk today about our work on computational approaches to study human immunogenetic diversity. So I hope everyone can see the slides. I think questions can be written in the chat afterwards. And I would like to start with a very broad introduction into the topic of what we are trying to achieve in our group. In this slide, so it's a very broad intro to how immunotherapies can work differently in different patients. So when a patient is given an immunotherapy, it's very difficult to predict how the patient would react, whether it would be beneficial or not beneficial. And this is in the particular case of cancer here, these two examples. So we see on the left side for an anti-PD-L1 treatment in colorectal cancer that patients may respond differently, and also on the right side in antigen-specific vaccination in brain tumors that the same happens. And this is something I personally find very interesting. I was already interested in this during my PhD when I worked on B-cell responses in malaria, and I always wanted to understand why it is so different in patients. So are there things that are similar between the patients that react well? Are there differences that you can observe between the ones where it does not work well and the ones where it works well? And this is also something that I would like to continue in my group now. Of course, there's a whole set of different reasons why there's so much heterogeneity, and the list I'm showing here is not comprehensive. So it can be the tumor phenotype, obviously, with signaling or metabolism. It can be the immune cell phenotype, or it can be antigen specificity, and many of those more. And we are focusing particularly on the immune-tumor interactions in my group. And what we are trying to achieve is to generate a quantitative understanding of how the immune cells interact with each other and how the immune cells interact with the tumor cells inside the tumor. And this we do by analyzing molecular data, so multi-omics data generated from tumor tissue, from human tumors, and extracting information in order to build a quantitative model on how the cells interact. And these could be T cells, B cells, antibodies, but also NKs or other cells that are present in the tumor microenvironment. So this approach leads us to the research focus of my group, which can be defined at different levels, going from the immunogenetics, so how the immune genes or genes that are important for the immune system encode receptors and ligands that are necessary to mediate cellular interactions, and how then these cellular interactions mediate immune function in the tumor. We have two main research strategies, which is first the development of bioinformatic tools for human immunology, and we then also apply these tools and other tools in biological discovery projects in collaboration with clinicians or experimental biologists. So far for the research focus. This is a quick intro about the group. So we started a bit more than 1 year ago in October 21, and we are currently doing purely computational work. So everyone in the group is bioinformatician or computational biologist. We are located at a hospital in the neuropathology department, which means that a lot of our colleagues are clinicians and also obviously work with tumor-related research questions. And we're also affiliated with the Mildred Scheel Career Center in Frankfurt and the Frankfurt Cancer Institute. And this whole environment makes it a very good place to study tumors in collaboration with other colleagues. So far about the group, and now I would like to dive into the topic. So what I would like to discuss during the presentation today is rather located in the first series of projects that we have, which are more related to immunogenetics and receptor interactions. And I would like to present two software development projects where we have developed or are still in the progress of developing software. One for naming of HLA alleles or converting of names of HLA alleles, and the second for immunogenetics in single-cell RNA sequencing data. So I'm sorry, I want to drink something. So I'm going to start with the first one, the HLA allele names. And I like to start with this slide. I mean, in this audience, probably everyone would agree that there's a huge diversity on the structural level of the receptors that coordinate the antigen-specific immune responses. And we are going to talk about the MHC molecules now that present peptide antigens to T cells, which are seen here. And the peptide has to be bound in a peptide binding pocket on the surface of the MHC molecule. And so it's well established that there's a huge level of allelic diversity that contributes to the HLA structural diversity, which is necessary so that one human can present a whole library of different peptides on their MHC molecules, and also on a population scale, there's a large library of peptides that can be presented. And when you look at the variations that exist between the different HLA alleles, you see that a lot of them, here shown in red, are actually located in the peptide binding cleft of the MHC molecules, and that they also encode amino acid changes. So this would change the structure of the MHC molecule and allow the MHC molecule to present another set of peptides. From a bioinformatics point of view, this is challenging because there's a lot of, tons of names, obviously, for the alleles. And if you, in a typical scenario, for example, you have a large data set of RNA sequencing data from a large cohort of patients, you perform HLA allele typing from the RNA sequencing data using a HLA allele typing tool, which works by aligning RNA-seq reads to HLA allele reference databases. And then you get results of HLA that are typed in the different donors, and this is how this might look like. So for HLA-A, you get 2 alleles, for B, for C, and of course for the MHC class 2 encoding HLAs, you also get different alleles that are annotated for each donor. And now, so I'm going to show 2 use cases of how you might want to continue from this stage where you have typed the HLA alleles. So the first use case is something that is frequently used in tumor-immunology. It's the prediction of the peptides that are presented on the MHC molecules using artificial intelligence-based tools that predict the peptides that are presented on a given HLA or MHC molecule. So it's the input format that you need for these prediction tools may vary. It's typically on protein level. And the challenge, bioinformatically speaking, is now to correctly convert the allele names that you got from the HLA allele typing to the protein level input format that is suitable for the prediction tool that you want to use. So as seen here, so you have a large cohort. You want the MHC complex names, and you want to predict what are the presented peptides. So this is use case 1. The second use case is a bit maybe more abstract. So in this case, you want to, you observe, you have two cells derived from 2 donors. And you observe a similar reaction to in terms of antigen-specific T cell activation. So there has to be something similar that is presented on both cell preparations. And it's important to note that the allele names that you find when you type the HLA alleles, they also differentiate between sequence variants that are outside of the peptide binding pocket. So you might have alleles that have different names, but that in the peptide binding pocket still have the same amino acid sequence. And the challenge here is then to find the HLA alleles with the same peptide binding pocket on amino acid level. So as depicted here, you want to basically know which could be, when you observe the same reaction, which could be the MHC molecule that is responsible for that. So these two use cases, I hope they speak to you, and maybe some of you have already performed these kind of studies and you're aware of the fact that it's sometimes can be very challenging to do this correctly and consistently with the HLA names. So this we were also in that situation, and this is why we developed a package we called Immunotation. So it's an R package that is also available in Bioconductor. And what it does is to interconvert between different naming schemes, so serotypes, the HLA alleles from RNA sequencing data. It also provides conversion of multiple allele codes. This is something you get back, for example, when you do HLA typing, for example, with a blood donation service, or other older HLA naming schemes. And then it provides conversion to the MHC complex names. It allows to generate input for the MHC binding prediction tools, and it also interlinks with a database of HLA alleles in worldwide human populations. So the backbone of Immunotation is built on an ontology, which is the MHC restriction ontology. It's the same ontology that is also used by the immune epitope database, IEB. And this MHC restriction ontology, it solidly links the names of the MHC complex to alleles, genes, loci. It uses the standard HLA nomenclature, and it also provides this for different species. Of course, in our most of our project, we are only interested in doing it in humans, but in case someone is also working with mouse, it's the same principle. And it allows you to link the different levels of information consistently. In addition to the MRO resource, Immunotation is also using the IDP-IMGT HLA database. As I mentioned, it's using the allele frequency net database for the worldwide population, and it's using MAC conversion tools for the multiple allele codes. So now as one example, what you can do when you combine all the functionality of the tool. So what you see here, it's an analysis that is done using the different functions of Immunotation package. And it answers the question, so for which human populations can we currently predict HLA presented peptide? And it's focusing here on only HLA presented peptides. And so you see something that might not be too surprising. So the higher the coverage here, the better the prediction, because the more alleles that are present in the population are covered by current prediction tools. And not surprising, there are a lot of European, North American, and Asian population where it works sufficiently well, but there's a whole area of the globe that is not covered. So there's still a lot of work to do to get this working worldwide. And it's hopefully also something I will see in the next years, that this gets easier. For example, when working with infectious diseases in different human populations. So far for the Immunotation package. Sorry, I'm going to drink something again. Now I want to get to the second part of the presentation, and this is again a software development project which deals with immunogenetics in single-cell RNA sequencing data. So here, the situation gets even a bit more complex in terms of immunogenetic diversity. And so this slide is summarizing the characteristics of human immunogenetic diversity that I'm going to focus on in the next talk that are going to be important. And they can be summarized by polygyny. So this means that multiple similar genes encode same or similar function. This is the case, of course, for HLA genes, particularly of the class 2 type, where there's also a combination of different genes that come together to combine complexes of MHCs. And it's also the case, for example, in the light chain constant loci of the immunoglobulin locus, where multiple constant genes can encode the same functional protein subunit, which is the light chain of an antibody molecule. So this is polygyny. The second one is hyperpolymorphy, which we already covered a bit in the first project, which means that there are multiple alleles for one gene. And this you've seen for the HLA molecules. So these two, and I'm focusing here on the most important loci, which I believe are important for the project, which are the IG, the TCR, the KIR, the HLA, and the MYC loci. And so for the polygyny, I tried to summarize this in the tables here to give you an example. So it's non-comprehensive, but so that you know what I'm referring to. So for IgG, for example, the number of similar genes, including pseudogenes that are known, and this is relatively recent version of the IMGT database. It's 5, and for HLA-DRB, for example, it's 8. So you know what I'm talking about here. And for the number of alleles, it's much higher, obviously. So for for the constant G gene, it's 71, and for HLA-DRB, it's around 4,200, so much more. And this is probably, the real numbers are probably even higher because a lot of the alleles are not in the database, as we've also seen in recent work by people in the community. So from here, let's quickly look at how typically transcriptomic analysis workflow works. So this is a depiction of so what is the typical procedure when you have transcriptomic data. So you want to go from the transcriptomic to generate single cell or spatial transcriptomic data or bulk sequencing data. You perform the data analysis or statistics, and then you interpret in order to draw conclusions about the immune function of the tissue that you have measured. And the experimental techniques that I'm talking about here and that we're focusing on are single cell transcriptomics, which comes in different flavors, so whole transcriptome from different sites of the transcript, or amplicon based, and also spatial transcriptomics. And the general idea of the data pre-processing is to quantify the expression of the transcripts in cells or at specific locations. So basically, from a computational point of view, single-cell transcriptomics and spatial transcriptomics are not so different because you quantify transcripts at a specific barcode, which can be cell or location. So let's look at the computational part of this in a bit more detail. So this is the overview of a typical computational workflow for the analysis and annotation of the transcriptomic data. And these are the different steps. So in the first step, you identify for each sequencing read the cellular barcode or spatial barcode. You identify the unique molecular identifier, which is basically the barcode that labels given mRNA molecule. And then you map the reads to a reference genome, and the features that are part of the reference genome are the genes. So you want to count the genes or the transcripts. And then after the alignment or the mapping of the reads to the reference, you build a count by counting the unique transcripts per gene and barcode using the UMI. From this, you then generate a feature count matrix, which has features times cells. This is also displayed here. So you have, this is how the bioconductor format looks like. So this is the cells are in columns and the genes are in rows. And the feature count matrix is this one where you count the features per cell. And then using this matrix, you perform the downstream analysis, which is dimension reduction, clustering, differential gene expression, everything that you need in order to answer your biological question. And now for the project, I would like to focus on this particular step here, how the reads are mapped to the reference genome. And now with the elements of the human immunogenetic diversity that we've collected in the previous slides of the talk, we know that there are some bioinformatics challenges that arise from one polygyny and also from hyperpolymorphy. These challenges are that so multiple genes or features have the same biological function, and especially when you perform downstream analysis like dimension reduction or clustering, you might be interested in the function and not the actual feature, because several features may encode the same function. The second one is problem what happens is multi-mapping, which is majorly due to the effect of polygyny, which means that there's a non-unique mapping of reads to genes when the genes with a similar function are too similar to each other, and the algorithm does not know which of the genes the read belongs to. And then there's another problem that arises, which is a combination of polygyny and hyperpolymorphy. This is cross-mapping, and it means that there's a unique mapping, but unfortunately to the wrong gene. And I'm going to go into a bit more detail about the last two, so about multi-mapping and cross-mapping now. So let's start with multi-mapping. So multi-mapping, again, it's the non-unique mapping of reads to genes. So this can happen when you have the lambda constant locus, and you have, so this is a group of reads belonging to a particular UMI group where the reads are clearly assigned to ID lambda C2. This is another one where the reads are clearly assigned to ID lambda C3, so this is non-problematic. But the green ones here, they are problematic because it's not clear to which of the genes they need to be mapped to, and this is why they are discarded in the current way how single-cell sequencing data or generally transcriptomic data is analyzed. This is how this looks like in real life. So this is a data set from human lymph node. And you see that everything in red has one unique mapping, but there are things in green here, for example, where there's a multi-mapping that is occurring, and you don't know whether they belong to ID lambda C2 and ID lambda C3. And if you look at the distribution now of how the curve of the counts per, so this is the position in the read alignment, how this curve of counts per read alignment looks like, you would probably guess that these reads actually belong to lambda C3 gene because they fit better on the curve here. But anyway, they are discarded in the current workflows. So we also performed a simulation to see how this may look when different donors have different alleles. So this is what you see on the right side now. So what we see is each column is the identity of the expressed ID lambda C3 allele, so lambda C3, O2, O3, and O4. And then the percentage of reads that are mapped to lambda C3 uniquely, which is in red, and non-uniquely in gray. And you see that so the number of non-uniquely mapped reads depends, one, on the actual allele of lambda C3 that is encoded, and also on the read length that you use in your sequencing approach. So if you use a read length of 76 compared to 48 base pairs, you get less cross-mapping, which is also expected because the reads can have a higher chance to be uniquely mapped. So as I said, the typical problem is that analysis tools discard the multi-mapped reads. And as we see in the simulation, in some cases where things are really in a bad constellation, there can be up to 80% of the reads that are lost. And what is particularly annoying when you want to compare multiple donors is that the frequency of the multi-mapping depends on the allele. So this means that there's a donor-specific quantification bias, and this reduces the inter-donor comparability. So far about multi-mapping. Now let's go to cross-mapping, which was the third phenomenon. So the third bioinformatic challenge that we discussed before. Cross-mapping is unique mapping, but to the wrong gene. And this is something that was actually surprising for us at the beginning, and it took us some time to understand how this arises. And this is a figure where I explain how this can happen. So again, it's so you have this typical problem, so gene A1, gene A2, which are very similar in sequence, and then transcripts that come from gene A1 and transcripts that come from gene A2. And then what is done in Cell Ranger, for example, so this is a typical quantification software, is that so the reads are mapped, and then the software checks to which of the genes are they mapped. And if they are mapped to multiple genes, the gene with a higher read count is counted. So in this case, for example, you have 4 reads to gene A1, 2 reads to gene A2, which means that gene A1 gets +1 count. But it might also be the opposite. So if you have more genes, more reads for gene A2, gene A2 might get a count, and the rest of reads is discarded. And so what we do here first on the right side is to look at, so which are the genes that are discarded, the reads that are discarded, sorry. And in the results file of Cell Ranger, these reads get a particular flag, which makes them easy to find. So we can basically tell why were they discarded and which genes were they mapped to. And we see here, so if we look at the on the x-axis, the number of cross-mapped reads, and on the y-axis, the total UMI counts in the sample for a particular gene. So each dot is one gene. So there's the majority of genes which follows a linear relationship, but there are some outliers here where the number of cross-mapped reads is much higher than expected from the distribution of the overall genes. And in red, we see the genes encoded in the immune loci that I was referring to, and you see that there's a clear enrichment of those genes in the genes that suffer from the cross-mapping. So what this cross-mapping leads to is that non-expressed genes seem to be expressed, and it's also allele dependent, which means again that intra-donor comparability is impaired. So for the ones who want to dive even deeper into this, sorry, you can do the same analysis, obviously, for different donors. And as you see here, so this is donor 1, donor 2, and you see the phenomenon stays the same. So there's an enrichment of cross-mapping in the genes that are important for immune interactions. But the pattern is different from donor to donor. So this is what I mean with a reduced intra-donor comparability. In addition, so let's dive deeper into this. This is now looking at UMI level. So we are looking now at reads that were mapped to one UMI, so basically referring to one original mRNA molecule. And you see that, so I can split the reads that are mapped to this one UMI according to whether they have unique mapping. So this is the first column, they have 2 mappings, or they have more than 2 mappings. And then in red, you see the ones that are mapped to lambda C2, and in green, the ones that are mapped to lambda C3. And you see that for this one UMI, we already have the problem that so the algorithm tells us this one mRNA molecule originated from lambda C2 and lambda C3, which is not really possible, biologically speaking. And in this case, so there's more blue than red, which means that this UMI would be counted as a blue count. So +1 for lambda C3. Now this, you can imagine if one cell contains multiple UMIs and you propagate this effect to the cell level, so this ratio may change. by random events. So here it's almost 50-50, but in some events it might be that the red ones are taking over. So if you propagate this to the cell level, this is what we are going to see here. So these are now reads from one cell. And so the ones that are counted that are neither multi-mapped nor cross-mapped are the ones that are here with the flag 25. So this is a flag that means that these reads are counted in the procedure. And again, we see that in this cell, according to the algorithm, both lambda C2 and lambda C3 are expressed. A tiny bit more of the blue one, which was also the one where we saw higher read counts in the UMI that we looked at before. And this is the problems that can arise in these. And if you're really interested in the biology of the B cells that you're looking at here, of course, these phenomena are something you don't want to see, and you need to correct for this somehow. And we also, so for this particular donor that we are looking at here, we also were able to show that the donor carries a non-standard lambda C3 allele, which is not referenced in the typical reference transcriptome or reference genome. So this is a problem here. So now I'm going to change gears a bit. So we have observed these phenomena, and in the lab we're working on different projects where we need to analyze single cell sequencing data and study immune cells. So we've developed an approach where we can quantify the allele-specific expression of particular genes, which has two positive sides. So you can avoid, to some extent, the quantification bias, and you can look at the allele-specific expression of a gene in a particular cell. This is the project of Ahmad, a PhD student in the group, and he ultimately wants to infer allele-specific interactions. And for this, of course, he also has to quantify the allele-specific immune gene expression. This is one example here from a dataset from a collaboration partner where we're able to quantify the expression. Here, these are HLA allele groups because we're not 100%. So the read coverage and read length does not allow 100% mapping of the HLA allele, but we can split them into groups. And you see that for particular donors, we're able to measure two different allele groups. So this has the benefit that you can now quantify on allele expression. And to take this further, so this is a project we work on with a collaboration partner, Federico Marini in Mainz. In this project, we take advantage of the allele-specific quantification in order to build a multi-layer data structure that can be imported into standard single-cell analysis workflows, where we can then simultaneously look at the allele level, the gene level, and the functional level. We're also working on building interactive data exploration tools for that. So this is where the project is going, and I would like to show you one example of how this multi-layer data integration can be useful when you want to study immune cells. Again, I'm going to show an example from B cells. So this is I'm illustrating how the multi-layer implementation is helpful to explore plasma cell transcriptomes. So this is a reanalysis of data from human colorectal cancer, focusing only on the plasma cells. And what typically happens, and maybe some of you have already seen in their own data, is that when you perform single cell analysis, dimension reduction and clustering, the cells start to cluster according to the constant chains, or sometimes also the variable genes that belong to the receptors that they express. So in this case, for example, you see, so here I show the expression of different genes, so lambda C3, lambda C2, kappa, constant, G1, and A1. And on the x-axis, you have the clusters, which are the same clusters as the single cell clusters that I've previously identified here. And you see that, for example, the green one would be the one that expresses lambda C3. The blue one would be the one that expresses lambda C2. And the D1 expressing ones would be the pink ones here in the middle. And so this might not be the best representation because maybe you're not interested whether the cells express C2 or C3, but you're interested in a more functional clustering of the cells. And this is something we propose now in our approach to aggregate. So this is rather simple idea, to aggregate the counts for the light chains and the heavy chains. And then use this as a functional feature count matrix that can be used to perform dimension reduction and clustering. And this is the result we obtain here. So you see that the different clusters that were dispersed before, they come together now and they form a they have a more continuous distribution in the expression space. And if you look, for example, at the expression of particular transcription factor, you see that you can better identify the cells that are positive for the transcription factors than you were able to do it in the analysis on the gene level. And so here's what's happening. So this is the table of how the clusters overlap. So in the functional level analysis, we have 4 clusters here on the y-axis. In the gene level analysis, we get 7 clusters. And the 4 clusters now summarize the clusters that were, or they incorporate the clusters that were previously separated. For example, the second cluster, get that right. Yeah, so the red ones here, for example, would be split in the second and fourth cluster of this one here. And the ones that were separated before, the third one, which is the lambda C2 cluster here, this would then spread across all functional clusters. So this is exactly what we were aiming in this approach. So this means that aggregating the genes on the functional level allows a more insightful clustering. And of course, in this multi-layer data structure, you don't throw away the gene level information or the allele level information, but you keep it in the same data structure and you can access it later on. So with this, I'm going to summarize and give a brief outlook. So I've, in the first part of the talk talked about the immunotation software package, which allows the conversion of HLA allele names for multiple purposes. It allows access to the database of human population HLA allele frequency. And I've illustrated 2 use cases for prediction of HLA presented peptides or for finding HLAs with similar function in a particular biological assay. The tool is available on Bioconductor. And in the second part of the talk, I illustrated how we approach the problems that arise through immunogenetic diversity in transcriptomic analysis. I showed you what multi and cross-mapping does with the data analysis. I showed you the benefits of using allele-specific quantification. And I illustrated how this multi-layer information can be useful to study cell phenotypes independent of their VDJ recombination or class switching history. So with this, I would like to thank everyone who was involved in generating the data that I've showed. I would like to thank my previous postdoc supervisor, who was very helpful in setting up some of the projects. People here in Frankfurt whom we collaborate on for single cell immunology. If you're interested in doing wet lab or clinical research projects, feel free to contact me. So we're currently recruiting here. And with this, I'm going to stop and I'm happy to take questions. I'm probably going to stop sharing.
Thanks, Katharina, for this.
I think I'm going to quickly turn on the light because it's getting dark here.
Okay. Yeah. Felix, you raised your hand.
Oh yeah, sorry, but I was confused there what function meant right at the end there. Like what were the different functions that were being when you were right at the end where you were saying you clustered by function instead of clustering by
I mean
constant region.
This depends totally on the biological question, right? So in our project, we often had the problem that the clusters that we observed, they were according to the gene expression and not to the function. So for example, I don't know, if you want to find all plasma cells, you're probably not interested whether which light chain they're expressing, but you want to have them in one combined cluster. This is what I mean. So and function can be as granular as you want it to be. Yeah.
Yeah, so some sort of cell phenotype function. Yeah.
Yes.
It isn't the suggestion. It wasn't that clustering by constant region was wrong. It just might not be what you were interested in.
Exactly. Yeah. And this is also why we preserve the information on the constant region in another layer of the data structure. Mm-hmm. So you want maybe afterwards, once you have identified all your plasma cells, you want to go back and look, I don't know, are there clusters of a particular light chain? And then you can go back, and so you can use the other feature count matrix to either perform additional clustering or perform differential gene expression analysis. So this is still possible.
And while I have you, so what is the representation of HLA alleles in the human? I mean, how you listed 20,000 alleles? Do you think that's getting a pretty good representation of the human diversity at that locus, or is it 3 times that, or does anyone have a clue? Or
I'm not going to venture and tell you a number now.
Yeah, I guess there's a lot of work that still needs to be done to quantify human variation.
Yes, definitely, and I think so, especially in populations that have been understudied so far. And it's definitely going to be more at the end. It's not going to be less.
Can you explain more about this multi-layer data structure?
So you mean how it's
multi-layer? Yeah, a bit more, maybe the computational.
Yeah. So basically, it's an object, and I mean, it's not a very complicated structure, but it's an object that keeps things together, and that has functions to address the different layers. And it currently implementation is under construction in the bioconductor framework. So using the single cell experiment class.
Nice. With regard to, so for the second part of the talk with the multi-mapping, all these kind of mapping things, what's the frequency of occurrence of these 3 different phenomena? This cross-mapping, Is one more often than the other? Or?
Yeah, that's a good question. So all of them are allele dependent. So it probably the real numbers depend on the donor. I would guess that there's more multi-mapping than cross-mapping. That's my impression. Yeah.
And does the discovery of those also depend differently on like sequencing depth and technology that one has used for sequencing?
Yes, definitely. So it depends on the length of the reads, which is a very trivial thing, right? It depends on the region that you've covered. So if it's an amplicon-based technique, of course, it might be less problematic. And of course, so, I mean, speaking of spatial transcriptomics, so everything I said only applies to things where you measure the actual RNA. There are a lot of new techniques now where that are hybridization based, where you don't sequence the actual RNA, but things that have hybridized to the RNA. And there, of course, none of this applies. So if it was developed, or if someone checked that there's no multi-mapping, yes. Yeah, this I haven't.
And yeah, I think my connection is again not really good, but yeah, but so with all these errors that can happen, what are the biological conclusions that can change on the, so what if we don't correct the data with the corrections that you propose?
Yeah. So expression levels between donors change because it's allele dependent. This we have, we've seen for HLA alleles that this starts to get, so you start to detect it because, so if you perform differential expression analysis, you start to detect the HLA alleles or the HLA genes. This is something we've seen. That's one point. And then, of course, this also affects the clustering and things that you see after dimension reduction. And I think originally this project developed because we saw in, I think this was lymph node data. We saw that it was very difficult to compare the expression profiles of the cells of the different donors because there was so much variability in the light chain expression. Yeah, but this, I think it was B-cell lymphoma, and this obviously the light chain expression plays a big role.
And I'm not so familiar with these constant region alleles and different constant genes. Is something about the differential biological function known? Because we know for example, V gene polymorphisms, they can have differential influenza binding or something like that for the B cells.
Yeah.
What's known on the biology of them? Yeah.
So for the light chains, I'm not 100% sure. I assume that there would. So we're probably at some point going to read that there are differences. Yeah, that's what I would guess now, but I'm not aware of any differences. I think for the heavy chain, so there are definitely differences between the different isotypes, even if there are only minor changes in the sequence. So in terms of Fc receptors, for example. Yeah, so.
Well, I mean, Nats or Gore might know too, but I mean, these constant regions are some of the ones that have some of the clearest disease associations, but then it turns out we don't really know the constant regions very well. And I wonder how much that, even in the databases, and they're not complete and stuff. That's what people have told me, but I haven't looked that carefully, but kind of scary.
It's definitely an interesting thing. And I would like to know more about it, but I can't answer your question right now.
Great. Any more questions from the audience? Then thanks, Katharina, for the very nice talk. And then we wait for 9 minutes for Jared's talk then. See you all in 9 minutes. Thank you. Thanks, Katharina.